Hello, welcome to part 2. Sorry, I had a little bit of trouble with my phone there. So we've talked about substrate level phosphorylation, and now we need to talk about oxidative phosphorylation. So most of the ATP produced in respiration takes place right at the very end in a process called the electron transport chain. Okay, and this is where most of the ADP is converted into ATP, is phosphorylated. And this energy comes from the electrons that have been moved around when we were talking about oxidation. Oxidation is the removal of electrons. So as we move electrons around, we actually release a little bit of energy, and that energy can be used to phosphorylate ADP. Okay, so this is why it's called oxidative phosphorylation, because it requires the oxidation of other molecules to provide the energy for phosphorylation. And we're going to come to that in detail when we have a look at the electron transport chain. So just to be aware, at the moment there are two types of phosphorylation: substrate level and oxidative. Decarboxylation, really easy, does what it says on the tin, takes away a carbon. Okay, interesting point. The carbon removed from glucose during the decarboxylation steps combine with oxygen to form carbon dioxide, which you all know is a waste product of respiration. Okay, so decarboxylation, removing a carbon. Another nice easy one, dehydrogenation, removing a hydrogen. Okay, this, like I said, dehydrogenation and oxidation are the same thing. So the hydrogens removed during a dehydrogenation process, those are, including their electrons, are what are carried to the electron transport chain that will give the electron transport chain the energy to phosphorylate ADP and produce ATP. Coenzymes, okay? These are all over our bodies. I mean, there's thousands of them. They're essential to life, but we only need to know about a couple of them in respiration. And basically, they help enzymes to do their job. Okay, they're not proteins, they're non-protein molecules, but they're essential to the function of certain enzymes, and they're essential to the function of the enzymes in respiration. And finally, electron carriers. So I've been banging on about oxidation and how it's the removal of electrons and how electrons have to be moved to the electron transport chain. So obviously, the things that carry these electrons are very, very important, and they're called electron carriers. So these molecules will accept electrons from the molecule that's been oxidized, so they'll become reduced, and then they will carry these electrons and pass them on to another molecule. It's a really, really clever, amazingly clever and essential part of respiration. So these electrons, like I said, once they get collected by the electron carriers, they get taken to the electron transport chain. And remember, the electron transport chain is where all of or most of the ADP is phosphorylated. Okay, just take a deep breath here. Pause me if you need to, because we're going to have a look at the new molecules. Okay, so I've introduced you to new terms, and we just got a couple of new molecules that we need to know about. So if we start off with the electron carriers, this can get a little bit tricky. Okay, so take your time, just have a think about it. Don't worry too much if it's not really clear yet, because until you actually see what electron carriers are doing in respiration, it's difficult to visualize sort of what their role is. You need to see them in action to really understand it. But just to give you a little bit of background, that's all I'm trying to do here. So the electron carriers in respiration are NAD and FAD. There's another one, NADP, but that's in photosynthesis, and we don't have to worry about that yet. So NAD stands for nicotinamide adenine dinucleotide, and FAD stands for flavin adenine dinucleotide. You do not need to know the long names. NAD and FAD are all you need to know. But the big names help when we're having a look at the molecule, because if you look carefully at this, kind of looks a little familiar. Okay, it's made up of nucleotide, pentose sugar, and phosphate molecules. Okay, does that sound familiar? So it fits in sort of like with this group of molecules that includes ADP, ATP, DNA, RNA. Okay, so it fits in with that group a little bit different. Okay, it's got two nucleotides, two bases, adenine, which we've heard of before, and nicotinamide. Okay, again, you don't need to worry too much about that. It's just for a little bit of background interest. Both of the nucleotides in this molecule are attached to a pentose sugar, as you can see that from the arrows that I've put on the PowerPoint. And these two nucleotides are joined by the phosphate groups. This is color coded, by the way. So if you have a good look, all the sort of like the keywords are the same color as the boxes. If any of you are colorblind and this is causing you a problem, please let me know and I will change the colors or take the colors away for you. Okay. So obviously, if NAD is an electron carrier, it can exist in an oxidized or reduced form. It can exist with a hydrogen and electron or without a hydrogen and electron. And this is really, really important because that feature of this molecule is what allows it to be an electron carrier. The way I found this slide a few years ago, and I really, really like it. Think of NAD and FAD as like electron taxis, okay? Right, little note here. If you are fantastic at chemistry and you have got chemistry and you know, no worries at all, and you know, Mrs. Davis, you're acing Mrs. Davis's classes, this is going to horrify you, so don't look at this. If you're a biologist like me and chemistry gets a little bit scary every now and again, this is a really nice analogy for how NAD and FAD work. Okay, so NAD is like a taxi, okay? And these two people here, they are electrons and they're leaving, let's say they're leaving a pub, okay? And they need to get to the next pub. But they can't do that alone. It's too far, okay? Or they've had too many gins. I don't know, either way. So these electrons need to get from pub A to pub B. So what they do is they jump into a taxi. So the electrons move from one molecule into the NAD molecule because the NAD molecule can receive electrons. They then move along. The electrons get to the next pub or they get to the next molecule, whichever way you want to think about it, and then they're released. They get out of the taxi, they're released, and they can go into the next pub, or they can reduce the next molecule. They can move into the next molecule. So NAD and FAD carry electrons from the glucose molecule, which is gradually being disassembled and having all of its energy taken out of it, and they carry those electrons to the electron transport chain, where all the energy in all those electrons can be used to phosphorylate ADP. Okay, so NAD and FAD, electron taxis. Right, what I'm going to do now, okay, I'm going to stay here. But I'm going to play this video for you, and the very nice American man will explain to you in slightly more scientific terms than electron taxis how NAD works as an electron carrier.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme, and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Okay, so like I say, explaining NAD and FAD from a slightly different standpoint. This is a technique I use a lot, especially in slightly more difficult topics. I will give you a description in lots of different ways. I'll often give you 2 or 3 different videos, and that's deliberate because everybody learns in different ways. And everybody finds their own way to make sense of respiration. So the more resources that I can give you, the more chance you have of finding the way of explaining things that suits you. Okay, so if at any point I'm using an analogy or using a resource that doesn't help you, ignore it and move on to the next one. Find the one that works for you. Okay, moving on. Another new molecules, the coenzymes. Like I said, coenzymes are things that enzymes need to work. In respiration, they also work as carriers. So they carry molecules from one place to another. And coenzyme A is the main one that you need to know about. And it basically transfers the product of the second stage of respiration to the third stage of respiration. I'm not going to go into any more detail on that at the moment. Just be aware that coenzyme A is something that you're going to have to learn about. Finally, the enzymes. I've been through this before with my class, but enzymes are really quite useful because they generally tell you what they do. A's on the end of an enzyme, end of a word in biology, tells you that is an enzyme. It tells you that that's what that molecule does. So the suffix A's tells you it's an enzyme, and the prefix usually tells you what it does. So decarboxylase catalyses a reaction that decarboxylates or removes carbon. Dehydrogenase is an enzyme that catalyzes a reaction that removes hydrogen, dehydrogenates. And ATP synthase is an enzyme that catalyzes the synthesis of ATP. Okay? So when you're thinking about these enzymes, or if, for example, you've got an enzyme in an exam and you're thinking, oh, crikey, what is it? Take a step back, pause, have a look at the front part of the word and say, what is that? What's it telling me that's going on here? Okay, so you will be relieved to know that's the end of the new terms and new molecules section. Go over it again. I have left just the PowerPoint on so you can read this without having me blathering on in the background, or feel free to listen to me again. Have a read through Mr. Pollock's introduction to respiration. I've put a couple of videos on. Have a look at those. Okay, this is all in preparation for beginning to think about respiration. And I will probably put a quiz on towards the end of next week. But there's no real sort of deadline on this. I just want you to start sort of familiarising yourself with the whole process of respiration and what's going on with it, so that we can make really, really good use of this time that we've been given in this really unfortunate way, so that year 13 moves really smoothly and we've got loads of time to get everything finished. Okay. Last thing from me, remember I am always here, as is Mrs. Parry and Mr. Burke and Mr. Morgan. Okay, you can ask any of us questions about this. Just send us a message and we'll get back to you as soon as we can. And like I said, after half term, I'm going to try and set up some sort of video conferencing so that we can actually have face-to-face discussions if any of you have got any big questions for me. Okie dokie, take care until next time. Bye.